Transport across cell membrane. There are different types of processes through which the molecules or ions are transported across a cell membrane. They include simple diffusion, which is a non-carrier mediated process, active transport, and facilitated diffusion, and they are carrier mediated processes, and osmosis. Non-carrier mediated cell transport. They are the types of transport processes in which the molecules to be transported across the cell membrane does not require binding with the carrier protein in order to be transported across the cell membrane. Simple diffusion. It is defined as the movement of molecules or ions through the cell membrane openings or channels down the concentration gradient without binding to carrier protein. Now, if we divide this whole definition into different parts, it will show us or tell us the different characteristics of simple diffusion. Number one is that it involves the movement of molecules or ions through the membrane. Second, these opening or channels, these are the pathways that are followed by these molecules or ions in order to be transported through simple diffusion. Then down the concentration gradient, and no binding of carrier protein, like without binding to the carrier protein, which means that these molecules or ions, they do not require a carrier protein in order to be transported across the cell membrane. Okay, then what do we mean by this down the concentration gradient? Let's say this is a cell, and inside the cell we have 5 sodium ions, and outside the cell we have 2 sodium ions, which means that the concentration of sodium ions inside the cell is higher and concentration of sodium ion outside the cell is low. So the sodium ions will diffuse from inside the cell towards the outside of the cell, and this whole phenomena is called down the concentration gradient. Pathways of simple diffusion. So there are two types of pathways: interstices and water channels. Interstices are small intermolecular openings, and this pathway can only be followed by lipid soluble substances. Then water channels. These water channels are water spaces in the intrinsic proteins, and this pathway is followed by water or water soluble substances. This is a diagram of lipid bilayer, which was discussed in the previous video of cell membrane. We all know that in this lipid bilayer we have phospholipid molecules and the intrinsic proteins. Now these phospholipid molecules, they have small intermolecular spaces between them, and these intermolecular spaces are called interstices, and through these spaces or through these openings, the lipid soluble substances like carbon dioxide, oxygen, or steroid hormones will diffuse from extracellular space towards the intracellular space and from intracellular space to extracellular space. Now, why only lipid soluble substances? Because we know that this middle part or the interior part of the lipid bilayer consists of fatty acids, and because fatty acids are organic in nature and they're hydrophobic, so these fatty acids make this part of the lipid bilayer hydrophobic. So only lipid soluble substances can diffuse through the lipid bilayer through interstices, not hydrophilic substances, because they'll be repelled by this part of lipid bilayer. And we know that in the lipid bilayer we have these intrinsic proteins that are embedded in the lipid bilayer, and these intrinsic proteins have water channels that pass all the way through this protein, and they allow the diffusion or the movement of water or water soluble substances from extracellular space to intracellular and from intracellular space to extracellular space. Calculation of diffusion. For calculation of diffusion, we have an equation which is J is equals to minus PA C1 minus C2. This J in the equation, it represents flow or flux, and it is the measurement of amount of substances that will be diffusing through the membrane per second, and the unit of flow or flux is millimole per second. Millimole is the amount of substances diffusing per second. This negative sign, it shows the direction of diffusion, which we know that it is downhill or down the concentration gradient, from high concentration towards the lower concentration. This P represents permeability, which is the measurement or the measure of ease with which solute particles diffuse through the membrane, and the unit of permeability is centimeters per second. This A is the area of membrane which is involved in the diffusion process, and its unit is centimeters per square. The C1 and C2 are the different concentrations across the cell membrane, whereas C1 minus C2 is the concentration difference across the cell membrane. From the equation of diffusion, we can predict certain characteristics of diffusion. J is directly proportional to P. With increase in permeability, the number of molecules or ions diffusing through the membrane, which is the flux, will also increase, resulting in increased diffusion. The J is directly proportional to A. With increase in area, the flux will also increase, resulting in increased diffusion. This plane A have greater area as compared to B, so number of molecules striking this plane A will be higher as compared to B, which means that when the area is greater, the number of molecules diffusing through membrane will be higher as compared to smaller area. So diffusion through A will be higher than diffusion through B. J is also directly proportional to concentration difference. Greater the concentration difference, greater will be flux and greater will be diffusion. As we can see in this diagram, that number of molecules inside the cell are less and molecules outside the cell are more. So there is a concentration difference. A number of molecules diffusing inside the cell from high concentration to lower concentration depends on the number of molecules that are present outside. So greater the number of molecules or higher the concentration outside the cell, greater will be diffusion inside the cell. So this means that greater the concentration difference, greater will be flux and so the diffusion. And this negative sign, it shows us that diffusion is downhill, which means down the concentration gradient from high concentration to low concentration. Factors affecting permeability. This permeability is the ease with which solute particles will diffuse through the cell membrane, and it depends on certain factors like oil or water partition, coefficient of solute, radius or size of solute particles, membrane thickness, lipid solubility, and lipid insolubility. So what is this oil or water partition coefficient of solute? We know that the internal part of lipid bilayer is made up of fatty acids, and this part is organic in nature. So solubility or permeability of a solute particle depends on the solubility of that solute particle in this fatty acid part of the lipid bilayer. So more soluble that solute particle is in this part of the membrane, more will be its permeability across the cell membrane. And this property of solubility of solute particles in any organic solvents, or this central part of lipid bilayer, is given a unit of measurement which is called oil or water partition coefficient of solute. So more soluble the substance is, greater will be the partition coefficient and higher will be permeability across the cell membrane. Radius or size of solute particles. Smaller the radius, greater will be diffusion coefficient, greater will be speed of diffusion, and more will be the permeability across cell membrane. This diffusion coefficient is the measurement of how fast the solute particles are diffusing across the membrane. For example, this is solute 1 and this is solute 2. Its radius is greater than the radius of this particle. It is small in size, so it will be more lighter in weight, and its speed will be high. So permeability will also be high, and the diffusion as well. Membrane thickness. Thinner the membrane, less will be the distance those solute particles will be covering while passing through the membrane, and greater will be permeability. This is membrane 1 and this is the thickness of membrane 2. It is thinner and this is thicker. So the solute particles diffusing through this thinner membrane will be covering less distance compared to this one. Lipid solubility. So greater the lipid solubility of solute particles, greater will be permeability and greater will be its diffusion. Lipid insolubility. So greater is the lipid insolubility, less will be permeability. Since lipid insoluble substances have low permeability, so what are the factors that affect their passage or their transport across the membrane? We know that hydrophilic substances are transported through pores and through water channels. But what will be the factors that will affect the transport of these hydrophilic molecules or ions that have charge on them. Factors that affect the permeability or flux of ions are concentration difference across the cell membrane and potential difference across cell membrane. So what is this concentration difference across cell membrane? We have this container-like structure which is divided by a thin membrane. On A side we have more sodium ions and less chloride ions, and on B side we have more chloride ions and less sodium ions. So there is a concentration difference of the ions on A and B side. As a result of this concentration difference, sodium will diffuse from A to B, from higher to lower side, and chloride ions will diffuse from B to A, from greater to lower side. And what is this potential difference across cell membrane? We have this container divided by a thin membrane. On A side we have more chloride ions, and on B side we have less chloride ions, whereas sodium ions on both sides are same in number. Since chloride ions are greater in number, so A side of the membrane will become negative, and since chloride ions are less in number on B side, so this side of membrane will become positive. This difference in concentration of chloride ions across this membrane will create a voltage, and that is called potential difference. And when this same process, when it occurs in a cell, a potential difference will be created across cell membrane. Because of this difference in concentration of ions, the A side of the membrane is negative and B side is positive. This negative on this side will attract sodium ions that are positive. As a result of this potential difference, sodium ions will diffuse from B towards A. And when diffusion of these sodium ions from B to A, when continues for some time and enough sodium ions have diffused, so what happens that a concentration difference is created. Now sodium ions on A side will increase in concentration, and on B side the sodium ions will decrease in concentration, due to which a positive charge is created on A side of the membrane and a negative charge is created on B side. As a result, this whole process will reverse and sodium ions will start diffusing from A towards B until an equilibrium is achieved. Carrier mediated transport. It is a type of transport mechanism in which the molecules or ions have to combine with the carrier proteins so that they can be transported across the cell membrane. And the characteristics of carrier mediated transport are stereospecificity, saturation, and competition. Stereospecificity. Stereo here refers to stereoisomers, which are the two or more similar molecules that differ only in the spatial arrangement of atoms. D-glucose and L-glucose are the stereoisomers. They are similar in structure. The only difference is the arrangement of hydroxyl group at carbon number 5. In D-glucose, this hydroxyl group is on the right side of the structure. whereas in L-glucose this hydroxyl group is on the left side. This is the only difference that they have. And the carrier-mediated transport mechanisms will differentiate between these two isomers, the stereoisomers, whereas non-carrier mediated will not differentiate, such as facilitated diffusion will transport the D-glucose but it will not transport the L-glucose. Why? Because it can differentiate between these two structures, because it is carrier mediated and every carrier is specific for each and every solute particle or any molecule, whereas simple diffusion will not differentiate because there is no carrier, it is not specific. So this ability of or property of differentiating between the stereoisomers is called stereospecificity. Saturation. In carrier-mediated transport, the transport rate will increase with increase in solute particles until carriers are saturated. Competition. The structurally related solute particles will compete for the receptor sites. These are structurally related solute particles. For example, they will compete with each other for the receptor sites. Facilitated diffusion. It is defined as the movement of molecular ions through cell membrane down the concentration gradient in combination with carrier proteins and without utilization of energy. If we divide this definition into different parts, it will show us the different characteristics of facilitated diffusion. Number one is there is transport of molecules or ions. Second is that for the transport, these molecules or ions have to combine with the carrier protein. Third is that there is no use of energy. There is no use of ATP when this facilitated diffusion occurs. And fourth is that it is downhill, down the concentration gradient. The process of facilitated diffusion. What happens is that the molecule to be transported enters into this channel through pore and opening. It travels downward through this channel, chemically binds to the receptor as seen in this diagram. The moment this occurs, within fraction of second, conformational changes occur in this carrier protein as seen in this diagram. When you compare the structure of these two and the structure of this protein, so they are quite different from each other. The chemical bond between the molecule and the receptor site is very weak. It breaks down due to thermal motion of this molecule. The opening on the intracellular side of this carrier protein opens and the molecule is released into the intracellular space. Example of facilitated diffusion is that the transport of glucose into muscle and adipose cells occur through this facilitated diffusion. In diabetes mellitus, there is no transport of glucose into adipose and muscle cells because there is no insulin. The role of insulin here is that it activates the carrier proteins which are required for the facilitated diffusion of glucose into these cells. Since there is no insulin, so there will be no activation of those carrier proteins, and eventually there'd be no transport of glucose into cells, and its level rises in the blood, resulting in this condition which is called diabetes mellitus. This table is comparison between the simple and facilitated diffusion. Concentration gradient is downhill in both of the processes. There's no use of metabolic energy. Both are passive. Facilitated diffusion is carrier mediated, simple is not. The rate of transport is directly proportional to concentration in facilitated diffusion. Facilitated is rapid, simple is slow. There is no inhibition of sodium potassium pump by both of these processes, whereas stereospecificity, saturation, and competition are the features of facilitated diffusion and not simple diffusion.